Our next speaker today is Anice Lowen.
So for those of you who aren't familiar with reassortment, I'll start by defining it. It's a form of recombination, which is specific to viruses that have a segmented genome structure. So influenza A viruses have a genome with 8 RNA segments. And this allows a very facile form of genetic exchange. When 2 viruses infect the same cell, intact gene segments can swap between viruses, giving rise to chimeric progeny genotypes in the progeny virus population. So that process is illustrated here in this schematic. Oh, pardon me. All right, so I'd included this image of a cartoon of a virion, really just to underline that point that the genome is segmented. We have the 8 pieces of RNA encoding a relatively simple set of viral gene products, just about 14 proteins encoded. And again, the fact that the genome comes in those 8 pieces allows reassortment to occur, giving an important source of viral genetic diversity. So influenza A viruses infect a wide range of host species, including avian and mammalian hosts. On this diagram, the natural hosts are shown, and the black arrows connecting them show documented instances of interspecies transmission. But those interspecies events are really relatively rare, and the norm is shown here with the colored arrows that I have put over top. Okay, so we typically have a given lineage of influenza A virus circulating in one species or a group of related species. And in that process, the virus will adapt to a given host species, such that there are host restrictions that limit interspecies transmission events. These host restrictions are really important because they are what stands between us and influenza pandemics. Unfortunately, these barriers can be overcome. We do have interspecies transmission events occasionally, and a subset of those can establish new lineages, and when that happens in humans, it is an influenza pandemic. Okay. So this slide is showing the origins of the last 3 influenza A virus pandemics in humans. So the first 2 on the slide in 1957 and 1968 had similar origins. These are both caused by reassortant influenza viruses that took some gene segments from an avian source, shown here in blue, and some from a human seasonal source, shown here in red. And so importantly, the spike glycoprotein of the virus, the hemagglutinin, in both cases was derived from an avian source. So this gives the antigenic novelty that allows the virus to cause a pandemic. Also importantly, the red segments here, the human ones, were already adapted to the human host, allowing transmission in humans to occur more readily. Okay? So reassortment can give sort of an evolutionary shortcut in the context of interspecies transmission events. In 2009, our most recent pandemic also involved reassortment, and it was a much more complex situation that played out in pigs over about a 10-year period preceding the pandemic. There were 4 viral lineages involved in this reassortment, and the very chimeric strain, pardon me, that formed in that process then transmitted directly from pigs to humans. All right, so I'm not sure how many slides I skipped, just one. Okay. All right, so I've told you that reassortment is an important source of viral genetic diversity. To put a number on that, if 2 viruses that differ in all 8 gene segments enter a cell together, 256 distinct genotypes can emerge from that cell. Okay, so you can have a pretty big burst in diversity just from a single co-infected cell. Now, it's important to remember that, like mutation, reassortment is typically deleterious. And this is because the chimeric viruses that form usually suffer functional mismatches among the components that have been brought together and have not had an opportunity to co-evolve. So hopefully, with the story about the pandemics, and I should have mentioned also that reassortment is very important in the evolution of seasonal influenza as well. And it really does make a tangible contribution to how these viruses evolve at a population level. So hopefully I've convinced you of the importance, and so surprisingly, when we started on this topic about 10 years ago, it was a really understudied area in the influenza field. And one fundamental question that hadn't been addressed is the one shown here. What is the efficiency of reassortment in a co-infected cell? Right, so I'm showing you on the slide two potential models that could play out within an infected cell. On the left, the idea is that the replicating viral genomes in the cell would be compartmentalized, limiting exchange of gene segments and probably supporting only low levels of reassortment. And then on the right, we could alternatively have a situation where the co-infecting viral genomes mix freely, which would support high levels of reassortment. So we wanted to differentiate these two possibilities, and to do so, we started out by quantifying reassortment in a cell culture model. So the first scale we're going to look at is a pretty small scale. All right, so to do this well, we needed to design a system where the progeny virus population would not be shaped by selection to allow us to have an unbiased measure of the frequency of reassortment. All right, so we developed what we call our wild-type VAIR system. It's simply a pair of very similar viruses that we use in combination. The wild-type strain has a partner, which is what we call the variant. And into the variant, we've introduced just a single synonymous mutation into each of the 8 gene segments to act as a genetic marker. In addition, we mark each virus at the protein level by inserting a peptide that can be recognized by antibodies. Okay, so these are different epitope tags in each strain. But because the two viruses are so similar, derived from the same strain background, we very much expect that all 256 progeny viruses that can arise through their reassortment will be of equivalent fitness and therefore can be detected in an unbiased way. All right, so we took the wild-type VAIR pairing and co-infected cultured cells here at a high multiplicity of infection of 10 infectious units per cell. And our goal in using that high MOI was to have a high opportunity for reassortment, and that essentially all cells would be co-infected. And what I'm showing you are genotype tables indicating the genotypes of progeny coming out of 3 cell culture dishes shown here. Okay. So the 8 viral gene segments are labeled here across the top. If you can see that, yeah. And each row in these tables corresponds to a single clonal isolate from the progeny virus population. So where it's all blue going across, this would be a parental virus of the VAIR type. And here in the middle of the tables, you see the reassortant viruses that have a mixture of red and blue segments incorporated. And so clearly under these very permissive conditions, reassortment occurred readily. Now to look at this more quantitatively, we titrated the dose of virus added to the cells, starting again at an MOI of 10 up here, but titrating down to an MOI of 0.1. And what's plotted here is the percentage of the progeny viruses with any reassortant genotype, here on the y-axis, and on the x-axis, the level of infection achieved in terms of the percentage of cells expressing viral antigen. And so clearly the frequency of reassortment declines as the levels of infection decline, and that's certainly expected since frequencies of co-infection would decrease with levels of infection. But the precise relationship we saw here was really surprising. What we expected to see would be a curve more like this. And this is a theoretical prediction that came from a simple computational simulation. Of course, this model has assumptions. One important assumption is that reassortment within a co-infected cell is perfectly efficient. Okay, so we assumed that there was good mixing in the cell. In addition, we assumed that all infected cells produce the same number of progeny viruses. We've actually spent years on this puzzle now, as you can see from the papers cited in the corner. And I'm not going to go into a lot of depth on it today because it's largely published work. But what we've found is that second assumption is essentially grossly incorrect. Okay? So I'm going to summarize the results just with this slide to say that our data do certainly support that there's free mixing of co-infecting viral genomes within cells. But that intracellular dynamic is not sufficient to account for the high levels of reassortment we see. In addition, we have strong evidence that there's a focusing of progeny production within multiply infected cells. So in other words, there's a strong benefit of multiple infection such that these co-infected cells produce many more progeny than singly infected cells. That in turn augments reassortment. All right, so I've told you that reassortment is highly efficient within a cell and within a population of cells. What about within a host? Of course, in considering the potential for reassortment to drive influenza virus evolution, it's really important to think about whether the dynamics of the infection within a host would be conducive to supporting cellular co-infection. Okay. So in particular, for within host dynamics to support reassortment, we expect that the virus would need to be both high density and well mixed within the host. So just to spell that out, I've got a few cartoons here. If the virus was low density within the host, co-infection would be relatively rare. This is essentially a low MOI situation. One way for a virus to achieve high density in a tissue is through localized spread, creating foci, localized areas that would have a high multiplicity of infection. But these foci would typically be clonal, okay? And so the likelihood of genetically distinct viruses co-infecting in this scenario is still low. So taken together then, for reassortment to occur, we really need the viral population to be both high density and well mixed within the host. All right, so to evaluate the extent to which that's happening in influenza virus infected animals, we evaluated reassortment in 3 mammalian hosts, guinea pigs, ferrets, and swine. So guinea pigs and ferrets are both well-established model hosts, particularly for human influenza. They're good models in that they're highly susceptible to infection. The virus replicates to high titers in the upper respiratory tract. We have efficient transmission to contacts. And in ferrets in particular, we see mild disease signs with many strains. Now, pigs, of course, are a natural host of influenza A viruses, and they're very interesting when thinking about reassortment for 2 reasons. So pigs are an important intermediate host between humans and non-human animals. And humans interface extensively with pigs in agricultural settings, and we do see the viruses transmitting back and forth between humans and pigs. The second reason is shown here in the graphic. Pigs are susceptible to multiple distinct lineages of influenza A viruses, including those from different host species. And this creates the potential for co-infection with distinct influenza A viruses and therefore reassortment. Okay. So pigs are fairly famous in the field for being a mixing vessel for influenza A virus reassortment. All right, so to evaluate the within host dynamics and the potential for reassortment in these species, we applied our wild type VAERS system, here going in vivo. And in particular, all of the animals were inoculated with a 1-to-1 mixture of wild type and VAERS viruses in this H1N1 strain background. From all 3 species, we collected nasal samples longitudinally. From the pigs and ferrets in particular, we also took tissues on day 3 and day 5 post-inoculation. From the ferrets, it was a nasal turbinate sample and then a single lung sample. And from the pigs, we did more in-depth sampling to give us greater spatial resolution. So as shown here in the picture, from the pigs, we had a nasal sample and then a sample from each of the 7 lung lobes to evaluate. All right, now then each of the samples was processed to collect 21 clonal isolates and genotype them, okay? So giving rise to raw data like you see down here. These are the genotype tables similar to what I showed you before. So again, each sample would give rise to a genotype table, and that's the raw data we're working with. I'm not going to show you any more genotype tables, but just wanted you to have a visual of what type of data is going into the analyses I will show you. All right, so these 3 plots are showing representatives of each of the 3 different species, just a single individual of each species. And what's plotted is genotype frequency against time, and each color represents a unique viral genotype. The blue wedge on the bottom is the VAERS parental genotype. So despite an effort to use a 1-to-1 combination of wild type and VAERS, we do see overrepresentation of the VAERS parent, so maybe we didn't hit 1-to-1 very well. The orange right above it is the wild type, and then the other stripes represent unique reassortants that were detected. All right, so clearly we have reassortment happening in all 3 contexts. To look at diversity more formally, we applied the Shannon-Wiener Diversity Index, and here are showing the results of that for all of the animals. So we have diversity against time. And on the guinea pig and ferret plots, you can see that there's dashed lines and solid lines. These represent two different doses. The higher dose is the dashed line. We didn't see a strong dose effect, so we typically just handled all the data together. And so another clear thing you can see from these graphs is that there's little temporal trend in terms of the diversity generated through reassortment. So for that reason, to compare across the three species, we just pooled all the data from all doses and all time points. And those comparisons are shown here. So on the left, we have richness, which is here the number of unique genotypes present in a sample. And on the right, Shannon-Wiener diversity, which takes into account both richness and evenness, the frequency of the genotypes detected. All right, so comparing across species here, we can clearly see that by both measures, pigs were lower than both guinea pigs and ferrets. Okay, so the richness and diversity arising through reassortment in the swine host was lower than that in these two model hosts. Keep in mind, this is just using the data from the nasal tract. Okay, so particularly at the nasal site, this is true. All right, now moving to the data from the tissues. So to understand the distribution of the co-infecting viruses and the potential to reassort at different anatomical locations, we looked at these tissue samples. And remember that wild type and VAER do differ a little bit at the protein level because we've introduced peptides into each virus that can be differentiated with antibodies. So here in these tissue sections, which are taken from the ferret nasal turbinate, we've stained for the wild type virus to turn it green, and we've stained for the VAER virus to turn it red. And then on the right here, we have a merged image where you can see in an orange color where those antigens coincide. Okay, so co-infected cells would be visible on the right. So we do see a lot of cellular co-infection in this tissue and in all areas where there was viral positivity. We see a lot of co-infection. So an obvious example is shown here with the white arrow. This cell is clearly double positive. The yellow arrow is showing a cell that appeared to only be positive for the VAER virus. This prevalence of cellular co-infection was also true in the lower respiratory tract of the ferrets. So I think you can see here really clearly, just visually, that the distribution of these two viruses is heavily overlapping. So it suggests a lot of potential for reassortment. So moving now back to the genetic analyses. Here we're using the pie charts to show the frequency of different genotypes detected in the different samples. Right? And what we have here is a nasal turbinate on the top row, and then right underneath it is the lung from the same animal. So ferret 11 here, nasal turbinate and lung are shown. We have the day 3 animals on top and the day 5 animals on the bottom. All right, so a few important takeaways. So again, the dark blue is showing the VAER parental virus, and the orange is the wild type, and the other colors are reassortants. So you can see that we detected a lot of reassortants in the ferrets. Also, if you compare the top row to the row right underneath it, I think you can appreciate visually that there's more reassortants in the nasal site than the lung. The nasal site appeared to be more rich and diverse than the lung in ferrets. Another key takeaway, which I think you can see is really pretty obvious to the eye, is how distinct the two sites were within a given animal. Okay, so we typically detected quite a different set of genotypes in the nose and the lung of a given ferret. All right, so to compare the lung and nasal sites directly, here we again pooled all the data across time points and doses, and simply used a paired t-test to evaluate whether richness and diversity differed significantly. So they do, and both are higher in the nasal site, suggesting there's something about the dynamic in that tissue which is more conducive to reassortment compared to the lung. All right, moving now to pigs. Again, we saw co-infection was really common within the tissues of the pigs. Also, this is a lung section from a pig. So we have wild type, VAER, and the merge. And again, here the white arrows would show examples of cells that were doubly positive for both viruses. Right here, we've arrayed the pie charts spatially according to where they were collected from the animals. So you can sort of see the spatial distribution of the genotypes that were detected. The top 3 animals, we did have a nasal sample available. And so that's what's here at the top. And then the other pie charts are shown on the lungs where they were derived from. All right, so a key thing to see here is that although reassortants are detected, almost all samples contained reassortants. The parental viruses tended to be predominant. Okay, so either the blue var or the wild type in orange here were typically predominant. Okay, so we would say that reassortment was pervasive and that it happened everywhere, but not extremely frequent. And then again here in the pigs, we saw a clear spatial heterogeneity. Okay, so you can see that a given lobe looks quite different in terms of the genotypes detected from a lobe right next door. So this spatial heterogeneity led us to hypothesize that a lack of mixing between lobes could be contributing to the relatively low diversity we are seeing. And so to test that idea, we computationally simulated a situation where there would be free mixing throughout the tissue. And so we took all the genotypes detected in a given pig and then randomly sorted them back into the 8 sampled locations. That simulation was run 1,000 times. And so the results from the simulation are shown here with the gray violins. That would be the distribution of results observed if there were free mixing. And then the colored data points are showing the observed results. All right, so with exceptions, you can see that the observed data lie typically in the lower half of the distribution, so supporting this idea that the compartmentalization within specific lobes leads to lower diversity at a given site. All right, now to more formally quantify and evaluate the extent of heterogeneity across sites, we applied a beta diversity metric. So beta diversity is frequently applied to quantify the degree of differentiation among biological communities. Here, the communities we're considering are viral populations in specific lung lobes of the pig. And we've applied a relative scale, so it varies from 0 to 1, with 0 indicating that the 2 communities would be the same in terms of the genotypes that were detected there, and 1 indicating that they're entirely distinct. All right, and beta diversity is applied in a pairwise manner, so here I'm showing you a matrix of results where we compare 2 sites at a time. So for example, in this lower left corner, we're looking at the beta diversity considering the intermediate lobe and the left apical lobe of this pig. The color scale shows you that the high beta diversities, the more distinct populations are shown with a darker blue coloring. You can see that the whole matrix is dark blue. Okay, so we did see that beta diversity within a pig was typically high, indicating that the individual populations were quite distinct. And I think a really nice way to appreciate this result is to nest that smaller matrix here in the yellow square within a larger matrix, which also makes the comparisons between pigs. Okay, so for example, here in the lower left corner, we're looking at the comparison of communities that were found in pig 1 versus pig 6. Of course, an individual pig is a spatially distinct unit, and we would not expect to see commonalities in those communities. And then, of course, then the beta diversity values are high between pigs. Right? So the key point I want to raise then is that the results within a pig are pretty comparable to the results between pigs. So really underlining the point that a lobe is acting as a spatially distinct unit, and that viral replication within swine is highly compartmentalized. All right, so I've got a few conclusions here for you, and then I'll follow that with just a handful of implications of these findings. So we see that viral dynamics in vivo do support cellular co-infection and reassortment. In ferrets, it was really interesting to see that the viral diversity generated through reassortment is higher in the upper respiratory tract compared to the lung. In pigs, across all sites, reassortment gave rise to more modest levels of diversity. In both animals where we collected the tissues, it was clear that reassortants that formed in a given location tended to remain at that site, such that within host viral populations comprised multiple distinct compartments. So one implication of these findings is that opportunity for reassortment within a host does appear to be extensive, and we therefore think that the diversity generated through reassortment is likely to play an important role in influenza A virus evolution. I introduced this idea that pigs are a mixing vessel for influenza A virus reassortment. It really draws on information from the field. We very much see that in agricultural settings. But I would just say that the role of the pig as a mixing vessel is maybe more likely explained by their permissiveness to infection with diverse influenza A viruses, rather than some property of pigs that makes them intrinsically conducive to supporting reassortment. And finally, we saw this very striking spatial compartmentalization within the host, which we think is likely to have really important implications for within-host selection and its efficiency, and also likely to shape onward transmission and the severity of bottlenecks between hosts. All right, so my last slide is super important because this has been a very collaborative project. The postdoc in my group who led this work is Dr. Ketki Ganti, did an outstanding job. We've been very fortunate to work with an Emory undergraduate named Max Bega, who led the quantitative analyses and the simulations I showed you. Those analyses were done in very close collaboration with Katja Cole. And then the animal challenge experiments, Daniel Perez led the ferret challenge studies at the University of Georgia, and the swine work was done at Kansas State University by the groups of Jürgen Richt and Wenjun Ma. I'd like to thank the NIH for funding, and thank you for your attention.
Thank you so much, Dr. Lowen, for that wonderful talk. I think we have time for maybe just one quick question, and then we'll head out for our quick break.
Thanks, Dr. Lowen. So my question was around, what do you think would happen if the pigs were infected sequentially, let's say after a period of developing illness from one virus and then infected with a second?
Yeah, so the question was about, here in our model, we've used simultaneous co-infection. Of course, in the real world, a sequential super infection scenario would probably be more likely. So we have looked at that experimentally in guinea pigs and find that within about a 12-hour time window, there's a lot of potential for productive superinfection and reassortment. So I would expect that also to be true in pigs. After that time window, the animal became refractory to the second infection. Okay, so the second virus just didn't get in. But also, thinking within the host about the dynamics of the first virus setting up shop in an earlier time frame, it actually increased reassortment early on to have the first virus more widespread within the animal. When the second virus came in, there was more potential for co-infection, is my explanation anyway.
Okay, actually, we have one from online.
Okay.
Thank you. So the question is from Pablo. I can't see your last name. Hi, great talk, thank you. Do you think that the higher reassortment in upper tracts and possibly smaller ferrets versus larger pigs has to do with genetic bottlenecks as they progress downward, or what may underlie that?
Yeah, thanks. That's a really interesting question, Pablo. So we see more reassortment, with a very small sample size here, in some smaller spaces. Okay, so in the ferret compared to the pig, and in the nose compared to the lung, we have higher reassortment. So it could be that the more limited surface area in the upper respiratory tract or in a smaller animal leads to higher density of viruses and more co-infection and reassortment. I think that's a pretty logical explanation, actually. We'd maybe just need more time and context to evaluate it within to be confident. And then the potential could bottlenecks be playing a role as the virus spreads through the host. So here it's important to think about how we inoculated the different animals. So the pigs actually got virus both intranasally and intratracheally, such that these wild-type and parental viruses should be distributed throughout the respiratory tract upon initial infection. The ferrets got a fairly large volume intranasally. So again, I think the inoculum virus would land in the lower respiratory tract pretty efficiently right at the time of inoculation. And then the diversity we're looking at, the reassortants need to form following inoculation. And so there I definitely think we're seeing bottlenecks in that, as I mentioned, the reassortant viruses tend to stay within the location where they're generated.
Thank you so much.
Thanks, everyone.